Hello, everyone. Thank you for coming to the first of the BCS computational tutorial series. Cory Shain is a postdoc in the F. Fedorenko lab. He's studying language in the brain. And throughout his research, he's been developing new methods for time series analysis. So today, he's going to be talking to us about that. And there will be a lab portion at the end of it, so make sure to download the data. You can come to us, Greta or I, if you're having issues with that. Great.
Thank you. Thanks, everybody, for coming. Yeah, just to clarify what to download, because it'd be useful to get this going while I kind of give the conceptual overview of the method. You can focus in the Google Doc, assuming you have access to it, on the section that says installation. So you just basically can, like, this command here, you can copy paste into your terminal window and run it in order to install the software. And then the sample data and models are at the link under sample data and models. Thanks again for coming. So today I'm going to talk about a method that I've been working on developing to help us understand the dynamics of time series, such as ones that we often need to analyze in brain and cognitive sciences. And so, as I said, I'm going to give kind of a conceptual overview of the approach and what kinds of problems it's designed to solve. And then we'll dive right into kind of applied problems and how you might use this to actually analyze your own data. But the approach I'm going to be advocating today is called, I'm calling it continuous time deconvolutional regression, or CDR. So we want to use, in this department, we want to use experiments to understand the mind, but the mind is a complex thing, and it has complex dynamics. So in my area, which is language, a lot of experiments will present participants in experiments with words or sentences in isolation that have been carefully constructed by the experimenters in order to test a hypothesis of interest. But the phenomenon that we're trying to generalize to from those designs is this. Like, what's the mind doing in these kinds of situations? The kind of conversational setting for which human language processing is adapted, comprehending like multi-participant conversations or dialogues in a noisy environment, planning our own linguistic responses, and then integrating the information that we receive through the language medium into our broader kind of conceptual model of the world. So I think that there's growing interest in the language sciences in studying language in the wild in this kind of setting. And I think that growing interest is also true of a range of subfields in cognitive and brain sciences, where we want to know kind of what brains and minds are doing in more naturalistic settings. So I think there's this kind of complementary and growing area of research that looks at settings like this. But to make that shift from careful control of the stimuli to a more naturalistic design, we end up kind of shifting the burden of experimenter effort from designing the stimuli to designing the analysis. We don't have control over what kind of stimulation participants are getting. We only have control over how we're trying to make inferences about their mental patterns on the basis of the data that we get in the experiment. And so we need good control of or implementation coding of the critical variables that we want to examine in a naturalistic setting. We also typically need strong controls for the kinds of confounds that we want to kind of rule out or kind of parcel variance away from. And then we need a good statistical model that's going to allow us to make good inferences about the underlying processes. And it's on that last point about the statistical model that I'm going to kind of focus today. So in practice, I think a lot of times in naturalistic designs, we end up analyzing data using models that make pretty strong simplifying assumptions. And this can have some consequences for the results of our analysis. So models with excessively simplistic assumptions, that is, assumptions that don't align well with the kind of true underlying cognitive processes, can lead to misleading conclusions because our results may be affected by kind of modeling artifacts due to poor fit between the model and actual brains. And they can also limit the scope of hypotheses that we can ask questions about. So for example, if we're assuming that a particular effect has a particular time course, then we can no longer ask questions about that time course. And then finally, the simplistic models can impede pattern discovery. So we can end up kind of short-circuiting our capacity to discover new patterns in the data that we wouldn't have seen if we used a kind of a less assumption-laden approach. So today I'm going to be focusing on kind of, I think, 4 key assumptions that are common in this domain in some combination, and then how we can relax some or all of them using the approach that I'm advocating. So the first assumption is what I'll call simple response functions, where by a response function I mean the function assumed by the statistical model to relate an event in the world or in the experiment to the response that it will evoke in our dependent measure, and the thing that we're kind of acquiring through the experiment, whether that's a behavioral kind of reaction time or a neural signature. But the simplicity kind of cashes out in different ways depending on which kind of experiment we're modeling. So in the language world, we'll often do naturalistic reading studies where we take a look at how long people spend looking at words while they're reading kind of stories or newspaper articles or what have you. So in that kind of setting, a common simplifying assumption about the response function would be that the properties of the word are the things that influence the response to that word and nothing else. But in reality, it may be the case that the response time at a particular word is influenced not only by that word, but by some aspect of the preceding context that we're not capturing well with our model. So that's one kind of simplifying assumption in a behavioral domain. For example, in a naturalistic MRI experiment, a simplifying assumption might be that the hemodynamic response that governs the way that blood oxygenation changes in response to neuronal activity, that's going to be some kind of fixed shape for all the voxels and all the participants that we are going to be looking at in our experiment. When in reality, we know that that's not true, that the shape of this function changes between individuals, between regions of the brain, and that it can differ between different kinds of effects as a function of their underlying time course. So like things that take longer to process can have a slower looking HRF than things that take less time. So that's one kind of simplifying assumption that we can make about the response function in the MRI domain. And then in like EEG, for example, we might epoch our data, so like kind of like chunk out segments of the time series that are associated with the onsets of property of the stimulus, and then average those in order to get an event-related potential, or ERP. And the assumption there is that, especially in a naturalistic setting, that those averages are a valid way of finding that event-related response, when in fact, that response could be influenced by other events that occurred in the context and that are being included in the average. So different assumptions we make about the nature of time can influence the models of these response functions that we obtain. But that's just one of the assumptions. That's kind of the primary assumption I'm going to focus on today. But there are a couple of others I think that are relevant as well. A very common assumption is that the variance of the response is constant. So we're going to have a constant spread of responses around the mean throughout the experiment. And if that's not true, then that can have a big impact on the results that we obtain, because we're going to be overly sensitive to points where we've underestimated the variance, and excessively insensitive to points where we've overestimated the variance. So changes in the actual distribution of the response and not just its value over time can have an influence on the results that we obtain. Another assumption we often make is that the response function is stationary, so that it applies the same way at every time point in the experiment, when in fact people adapt to experiments and their responses could be different at the beginning versus the end of the experiment. They could be different as a function of which context they're in when they're responding. And then finally, we tend to assume linear, mutually independent influences of the predictors in our model on the response, which may be true in some cases, but it's a strong assumption to make, especially given kind of like the growing success of, say, deep neural network models, which are the best existing simulations of many kinds of complex cognitive processes that we have currently in the domain of speech perception or visual perception. And one reason why those models appear to be so successful is precisely that they relax this assumption that they can flexibly integrate and combine properties of the environment in nonlinear ways. Again, today we're talking about this approach that I'm calling continuous time deconvolutional regression, or CDR. where the primary contribution is to relax assumption 1, but then I'm going to talk about some generalizations that can also relax the remaining 3 assumptions that I'm talking about here too. But the idea is that we can kind of get past the necessity for some of these simplifying assumptions if we simply allow the response function estimated by the model to have a continuous time definition. Yeah, so that's the assumption we're targeting here with CDR. So to kind of like give a basic kind of conceptual overview of the approach, I'll start with something that's not CDR. So imagine that we have this time series. We've got some stimuli, x, of different values, and they're kind of sequenced in time in this way. And then we measure some responses to those events, y. And so we want to use the values of x in order to generate a prediction for the corresponding values of y. So in this example, we're making the assumption that the value of x determines the corresponding value of y, the one that's kind of co-located in time, and no other aspects of the stimulus sequence affect y. So this might be like a model that would actually be used, for example, to study reading, where we might assume that like how surprising a word is in context, imagine that that's what x measures, governs how long a person spends reading a word, that would be what y measures. So as I talked about before, that may not be true. We may have this kind of like lingering or residual effects of information processing load from previous words or previous stimuli on the response that we obtain at the kind of magenta time point. And so a common way of relaxing this independence assumption is to just add in some regressors to some finite number of preceding words, allowing us to get kind of like temporally weighted effects. So here we're predicting y not just based on its corresponding surprisal, but also on how surprising the preceding word was, as well as the word before that. And so we can get this kind of like discrete function that says like the surprisal kind of jumps in this particular way. And this kind of model will allow us to estimate that from data. And so this approach is called many things depending on your field. So some will call it distributed lag regression, some will call it spillover, some will call it finite impulse response modeling. It's the same thing, but the crucial commonality between all of these approaches is that you have this indexical notion of time, that it kind of like progresses as a function of order, but not as a function of the actual time stamp of these events x and the responses y. So if the events x are equally spaced in time, that's no big deal. You can just easily map from the order of an event to its timestamp. But when we're talking about naturalistic events and naturalistic stimuli, we're often dealing with kind of sparse, irregularly spaced events. And that scenario is where these kinds of discrete time approaches really break down. So the idea being that, imagine again that we're modeling reading times here. There may be a difference in the amount of influence that a preceding word has on y as a function of how long ago that word was fixated. So if it was fixated 100 milliseconds ago, there may be more kind of residual processing going on than if it was fixated 3 seconds ago. But this approach erases all of that information. We're not looking at the variation in duration of the events x. We're just looking at when they occurred relative to each other, their relative order. So the idea behind CDR is to take that variation in time into account. So instead of just looking at when events in x occurred relative to each other, we actually look at their timestamps, as well as the timestamps with which the responses y were measured. So here, the kind of like the same 5 events that I've been kind of cartooning before are shown here on the real timeline along with their associated responses. And so instead of defining the response function as like a vector of discrete weights associated with particular points in time or particular positions in time, we're defining the response function as a continuous function that has some parameters that describe its shape. So that's plotted in green, where IRF is short for impulse response function. And so what this buys us is the opportunity to consider the joint influence of all the preceding stimuli in the experiment as a function of how long ago they occurred in time. So we can take the response function associated with each of these impulses, each of these events, and then use the response function to compute a convolution weight for that according to how long ago it occurred relative to the time point that we want to predict, say this magenta bar, y. Which means that our prediction for y is conditional on the entire experiment, not just on the kind of co-located event. And what's nice about this definition is that it really it makes no requirements as to how x and y are aligned or measured, right? So because of this continuous time definition, we can rejigger where these responses get sampled, and we can apply the exact same model. So by kind of separating out the ways that events occur and the ways that responses are measured over time, we can get this very flexible mapping between any two kind of synchronized time series. And I'll say at this point that like the IRF, so I'm all about relaxing assumptions. Here's kind of the motivation that I've given, but now we're assuming a new thing, which is like the functional family for the IRF. So we can assume something that's simple, like say like a Gaussian-shaped IRF that has like a location and a variance, and that would be a kind of a strong simplifying assumption. But the mere fact of being parametric, we still can have very expressive assumptions about what the shape of this thing will be. So we can assume a simple kernel, but we can also assume, say, like a sum of arbitrarily many Gaussian functions. So it winds up looking like, for example, like a kernel density estimator that can have arbitrary shape. And we actually, I'll show in a second, can drop in a neural network here and have it do the same work. So this assumption of an IRF, I think, or an IRF family, is not particularly constraining. At least it's less constraining than some of the assumptions that I'm using this in order to relax. So to kind of put this picture in a slightly different way, let's imagine this is our kind of naturalistic stimulus with irregularly spaced events that are going to trigger some response. We want to estimate what that response is. We can take that stimulus and we can measure multiple different kinds of responses to it and capture their effects using the same model. So we might take behavioral measures of the reading times associated with, say, if these stimuli are words in an experiment. So they're going to be spaced out in time in the same way, but we still can capture these kind of diffuse effects of words from the preceding context. But we can also run this in an fMRI context where we have these like kind of slow but regularly spaced scans that are taken by the scanner. And then we can map the expected influence of each stimulus on a given scan as a function of their distance and time without needing to make any assumptions like that the hemodynamic response has a particular shape. And then EEG or MEG, it's the same thing as MRI, it's just much more densely sampled. So, so here we can use CDR in order to estimate event-related fields or potentials associated with particular words in an experiment, but in a kind of data-driven way that doesn't make assumptions about, for example, the validity of averaging multiple epochs together in a setting where the events, where the responses to events are going to overlap. So the upshot of all this is that we have a unified statistical framework for analyzing these kinds of time series, regardless of the temporal structure of the stimuli and the responses. So the approach works. I've applied it to reading, a couple different types of reading experiments as well as fMRI. It consistently outperforms established approaches using linear mixed effects regression or generalized additive models in kind of predicting unseen responses. So it generalizes better than alternative approaches, which at least suggests that it's tapping into generalizable features of the true response function that are difficult to capture using other means. So that was all about assumption one, like relaxing the simplicity of the response function by defining it in continuous time. I'll now like briefly go over how we can use the same basic framework and just kind of like some light generalizations of it in order to relax some of the other assumptions. So assumption two, which is that the variance is constant, can be relaxed by using an approach called distributional regression, which is you just allow all of the parameters of the predictive distribution over responses to depend on properties of the stimuli, not just their values. So typically we're estimating the change in the expected value, the mean of a response as a function of the predictors that are thought to influence that response. But we can also look at the influence of those predictors on the spread, the variance of the response, in say a Gaussian predictive distribution over reading times or the bold signal or what have you. So the same basic framework applies. We just modeled all the parameters of whatever distributional assumptions we're making about how the response is distributed, which is nice. That can give us useful information that's otherwise difficult to obtain about how, for example, the surprisal of words might affect how much spread you get in the response to those words. And then assumptions 3 and 4, which are respectively that the response function is stationary and that the effects of predictors are linear and mutually independent, can be relaxed by dropping in a particular kind of deep neural network as the IRF that I showed in the earlier plot. And that network relaxes both of these assumptions by allowing predictors to interact with each other in arbitrary ways, to have nonlinear effects on the response, and to depend not only on the values of the predictors in the events, but also on their timestamps, and therefore change over time, as well as change as a function of the preceding context. So I'm kind of skipping how the network does that for the sake of time, but the upshot that I'm trying to get across is that gives us a pretty flexible model of time series, that we can kind of relax a lot of the assumptions that we've previously had to make while still getting an interpretable description of the system that we're trying to model. So those are the main, I think, advantages of the approach. So in a nutshell, what CDR does is, given a definition of the model, that is like which predictors we're going to use to predict the response, which responses we want to model, and what kinds of assumptions we're making about the functional family that we want to estimate. We're just going to use stochastic gradient descent in order to optimize a regularized likelihood function. That'll give us kind of the best fitting model that we can obtain using current state-of-the-art deep learning approaches or machine learning approaches to get the parameters of the response function that we care about. And then we can inspect those estimates and see kind of what they show us about the response. And then we can do statistical testing on them by evaluating them with respect to some null model of interest in order to see whether or not we get improvements from some aspect of the response function that we care about theoretically. So that was a kind of a brief overview. At this point, I'll pause for questions.
Do you have to use some of your data and put it aside to first come up with this estimate of your IRF?
Yes, you should. Yep. So we do assume relatively big data that we're going to try to partition into training and testing sets. Cool. Well, that's a good segue into the caveats. So that's what I'll start with next before everybody gets too excited about this approach. So yeah, it's a data-hungry approach compared to, say, linear regression. Exactly how data-hungry is an open question that I'm trying to figure out. Like how little data can we get away with in synthetic experiments and still get a pretty good estimate of the response. So hopefully in a few weeks, actually, I'll have some answers for anyone who's interested on that. But it definitely requires more data than more basic approaches that make stronger assumptions. And as a result, it's compute heavy. It can take, a model that might take seconds in a linear regression context could take hours or even days to train here. So that has implications for the kinds of response variables that you can model. So it's pretty common, for example, in MRI to do like voxel-wise modeling using linear regression, in which case you have thousands of distinct models that track the time courses of each individual voxel in the brain. That kind of approach is a lot harder to do here just because of the amount of computation necessary in order to estimate it. But I will say if you use the neural net version, the system can kind of take advantage of mutual information between voxels in order to like share weights and accelerate some of that training. So you actually can fit these in a multivariate setting where you're kind of simultaneously modeling all voxels. And that can, although that's still certainly more computationally intensive than linear regression voxel-wise modeling, it's feasible, at least for moderate data sets. But yeah, it takes more computing. The likelihood function is non-convex, so there's no way of guaranteeing, this is the case with any deep learning model, that you have found the best fitting model to the training set. And because the model is so powerful and flexible, that isn't probably the thing that you want to do anyway. Like what you want is the system that will generalize the best. So for that reason, the training set itself, so models fit to the training set, is not a particularly informative metric, because it could reflect overfitting if you don't have any way of diagnosing whether your model's effects generalize. So for that reason, I guess this left it for last, it is a good idea to hold out some data so you can use it for evaluation and statistical testing. And then the point that I skipped over here, uncertainty intervals in the model are approximate, using different approaches from variational Bayesian deep learning, we can get some idea of how certain the model is about its parameters, but the posteriors aren't going to be exact, and so they shouldn't be used for testing. Yeah, those are the major ones I've run into. Yeah. Well, yeah, so if you hold out data or if you do another experiment, then there is a gold standard. Like you have the set of stimulus events associated with data that your model wasn't trained on, and you also have the measured responses. And so you generate predictions using those stimuli for the measured responses, and then you compare how good the predictions are. Does that answer your question?
I don't know what's the real IRF.
Yeah, so the question was, how do we evaluate the model's estimates against a gold standard? Yeah, and so that we can't do. That's true, because we don't know what the true response function is. That's the case for any statistical model of an unknown response function like the brain, right? So no matter whether we're using CDR or something else, we don't know what the true response is. We're trying to make inferences about what that response is likely to be. And a good way of doing that is looking at model fit. So not comparing the IRF to some gold standard IRF, but instead comparing the predictions derived using the IRF to measured responses from an experiment. So that's the approach that I'm advocating. In synthetic data, we do know the gold response, so we can assume a particular response function, generate data using it, and then fit the model and see whether it recovers the true synthetic model. And there, the answer is yes. I don't have demonstrations of that here, but we're going to fit, those of you who have computers will fit to a synthetic data set, and you should see these, the true model emerging in the estimates. But yeah, as soon as we apply it to things that we don't already know about and are therefore scientifically interesting, then we have to use proxies like model fit in order to select models.
Limit to how sparse the sampling frequency can be relative to the frequency of the actual response?
Yeah, there is. That's a good question. Sorry, the question is there a limit to, correct me if I'm wrong, but is there a limit to how sparse the response sampling frequency can be relative to the predictors in order to, so the stimulus. So if you get a lot more stimulus events per unit time than you get responses, does that limit the inferences that you can make about the response function?
How you record the responses. Let's say Fmri has a slower or longer interval for each response.
Right.
Can this still work?
It does, but the sampling frequency of the response, I think independently of the sampling frequency or the frequency of events, will constrain the temporal resolution of the estimates that you can get. Yeah, so there's no miracle here that can inject greater temporal resolution into Fmri than it inherently has. So you can't go beyond its Nyquist frequency in terms of the amount of resolution that you can get about responses. So even with Fmri, so the response function is going to be dominated by hemodynamics. Differences in kind of cognitive time, underlying time courses at the neuronal level, won't really emerge clearly there unless they're very large enough to kind of shift the hemodynamic response in a measurable way. But a few hundred, dozens or hundreds of milliseconds here or there probably won't emerge in even these kinds of models of MRI just because the signal doesn't support their discovery. Does that answer your question? Okay.
The model fits one IRF for all types of for stimuli, right?
No, not necessarily. Yeah. So you get to decide that as the user, how you want to tie weights in the model. Yeah. Tamar?
Try to test in a simple Fmri data set with a long interval between stimuli that it recovers the hemodynamic fold signal, assumed function. Is it similar?
No. I've only applied it to naturalistic Fmri, but it recovers something that looks like the canonical HRF there. And then
method leads to the same kind of assumptions that classical Fmri do.
Yeah, I mean, I think it does, because with a very like a neural net versus a relatively assumption-free model, you get the HRF from naturalistic data, even without spacing things out. So even with like compressed and overlapping responses, the same basic response profile emerges. But then, yeah, I've tested in synthetic settings with a bunch of different configurations, and it consistently finds a good approximation of the true model there. So I think between those two findings,
the real, if the assumption is the correct one in the fMRI, in the brain.
Yeah.
Yeah.
Yeah. So I think the result is that the resulting estimates from applying it to naturalistic data do look like the HRF, but they still fit better than assuming the canonical HRF.
Question from online: how can we use CDR for analyzing naturalistic movie fMRI data where stimulus events are not explicitly defined?
Yeah, so this it's a great question, and I don't have to repeat it because everybody here heard it. Okay, so you need some sort of representation of events in order to do this kind of modeling. So you need kind of like 2 fixed points and 1 unknown. So in this case, the assumption is that the fixed points are the measured responses and some representation of the event sequence. And then the question is, what function mediates between those 2 things? And we're going to use deep learning or machine learning in order to get an estimate of that. If you know neither the event sequence nor the response function, then you don't have enough information to fit this model. I think that in practice, it's often possible to come up with some sort of coding of movies or other naturalistic stimuli that could allow you to at least fit responses to kind of like gross, easily discoverable events in the sequence. But yeah, without that, there's not much that can be done here because the response functions are by definition responses to events. So it needs to know what's going on underlyingly. Move on to pitfalls, which I'm distinguishing from caveats, is like these are things that are easy to forget and mess up if you're used to like a linear or really any kind of like existing analysis pipeline. And this is a combination of like my own experiences as well as experiences of others who have tried to pick this up, and I realized, oh wow, that wasn't clear. So one thing, in some domains this is kind of obvious, in others it's less so, is that the predictors and responses are different things. They're distinct time series in this approach. So if you're doing like neuroimaging, this is an obvious thing. Like your predictors are going to be however your events in the experiment are coded, and your responses are going to be fMRI or like EEG measures. But when you're using, I think this becomes an issue in behavioral data when you're, for example, modeling like reading times where there may be just kind of one data set that represents the time series, and it includes a column for your response variable, but also columns for your predictor variables. And so it's important to remember that the two quantities are actually represented differently by the model. And this has implications for this point. I did this in the wrong order, which is not dropping predictors. So it's common to filter your data according to some sort of like outlier detection algorithms. And that's valid in this approach if you're applying it to the responses. So I'm not going to model the response in this chunk of the time series because I don't think that it's a good chunk for whatever, according to whatever criterion I've defined. But you shouldn't do that to the predictor sequence because then you're kind of telling the model that whatever happened here didn't happen. Those are the events that the responses are supposed to be associated with. So for example, if you're running this on a reading experiment and you're like, I want to drop all fixations that are longer than such and such. If you drop that from the predictors, then you're going to wind up distorting your model. So you just keep in mind that the predictors and the responses are different things. And that any sort of data censoring or partitioning, also partitioning data into training and test sets and so on, that should only apply to the response, not to the predictors. A second kind of like intuitively obvious thing, but that's easy to run afoul of, is that everything has to have a timestamp. A lot of times, people who collect data, especially like open data, haven't done this because they weren't planning on analyzing things in this way. But using a continuous time model, you need to know when things happened on the real timeline. And if you can't like either get or reconstruct that information, then you can't use this approach, unfortunately. So that's kind of, I've run into that. It's constrained my ability to use certain public data sets that don't have that information. You need to kind of think about what constitutes a run. Like basically like where am I going to chunk up the time series such that parts of the time series are treated as interdependent versus independent. For example, in like fMRI, this would probably be just corresponding to like an fMRI run. So like I'm going to have a time series associated with each distinct time a participant sat down in the scanner and stayed still and got scanned. But yeah, so you got to keep in mind like where the events are gonna, or where the time series themselves are gonna be kind of chunked up, because in a typical experiment you'll have, your table will represent multiple concatenated time series, and you want to avoid either spilling over into like the wrong time series, things that aren't actually consecutive in time, or chunking things up too much, where things that really are underlyingly part of the same kind of experimental like setup and time series are treated as independent when they're not, when they may not be, in fact. And then, as we've talked about, it's important to hold out some data for evaluation. Oh yeah, choosing a reasonable time scale. This one I've run into recently. So a lot of times, like sometimes experimental software will timestamp events as the number of milliseconds elapsed since January 1st, 1970, or whatever, like default representation of time. Which results in like huge numbers. And because this approach just assumes 32-bit floating point, like underlyingly, you can actually wind up with really weird degeneracies that you don't expect. So just in general, try to avoid huge numbers in your data frames. This often isn't an issue if you're just doing linear regressions in R, but it can be here. So I recommend like starting your time series timestamps as with 0 indicating the beginning of the run, rather than the beginning corresponding to, for example, midnight on January 1st in 1970. OK, so that's the end of my overview. Yeah?
In terms of the held out data, does that have to be like cross-validated? Or is it always like you leave something out and then that's always held out? And then second thing, what about in terms of like circularity, if you do some like downstream cross-validation analyses for like a different purpose?
Yeah, it's a great question. I mean, I don't think there's an inherent answer. You can do whatever makes the most sense for your needs as an experimenter. The challenge with cross-validation is that you can't use it for both tuning and testing. So one advantage and one reason why a lot of machine learning applications will have like a fixed like tuning and test set is so that they can try a bunch of different variants of their model on the tuning set, but then keep the test set like unsullied. So if you're not sure about your model definition, or you're worried about the influence of like some of the parameter settings for fitting, and you want to be able to optimize those before you do any evaluations, then I would not do cross-val. But if you're like 100% like you've got everything fixed in your mind, like this is, and you've like pre-registered, like these are the analyses that we'll do, then cross-validation makes more efficient use of your data.
Downstream cross-validation. So say that I modeled my data, and then I want to do like something else afterwards. Is that then weird that you use cross-validation to estimate the parameters, and then you, I don't know.
It can be. I think it depends on how different the something else is from what you already did. Yeah, I think it's difficult to answer in the abstract, but there's definitely more risk there than if you've got a special set. Yeah. Uh-huh.
I have a somewhat similar question. Out of curiosity, have you looked at, for example, let's say you're trying to model fMRI data. Is there decent like across data set activity? So for example, let's say that you have 2 different continuous naturalistic time series of words or something. You extract the same features from both. You have a data set, you train on that, test another one and collected, for example, day, but same person, same coverage. How does do you have any data on how that looks?
No, I don't. That's a great question. Yeah, so short summary is, have I evaluated how well model estimates generalize to new data sets from the same domain? Is that a fair summary? And the answer is not yet, but I've got something in the pipeline for that. So hopefully I'll know more soon.
Learning personalized HRFs, like if that would improve upon just assuming the canonical HRF scenario.
Yeah. Well, for that question, sorry, the question is whether learning a personalized HRF is better at describing the data than assuming a fixed like canonical HRF shape. And for that, I've shown that it is for at least one data set. But the question about whether the estimated data set works on a different data set that it should work for is a good one, and I haven't done that yet. If you haven't already, go ahead and install the software. And I actually think this slide is worse than just going to the tutorial page that was linked to in the email. So if you pull up the Google Doc, please somebody let me know if there's an issue accessing it. There's really two ways that you can install. I'm assuming that you all have Python installed, or Anaconda or something of that nature. So if you're an Anaconda user and you want to put this in an environment, there's instructions for doing so on the README. And if you just want to use kind of standard Python, you can do it with pip using this command. So again, this is to install an implementation of the framework that I've been describing. And this doc also has a link to sample data and models. So there's going to be two zip files that you want to download. One's called data.zip and one called models.zip. So you want to download those and extract them into a working directory that you want to use for this. It doesn't really matter where. And then the next thing is showing how to configure your experiment so that it can run. So yeah, so somewhere in your working directory you should have a models directory and a data directory. Depending on how your extractor worked, there may be like an additional nested level here, which we don't want. If you click on data and it just has another directory called data, then you want to like remove that. So move the lower directory into the higher one. Same with models. So this is what your models file should look like. ET stands for eye tracking, Fmri stands for Fmri, and SPR stands for self-paced reading.
Cory, are there ways to add like kind of meta-level breakthrough, like with the eye tracking, like end of line? Is that oftentimes something you'll see? Like regression? Okay.
Yeah, anything that you've got in your table can be used as a regressor. Yep. So CDR demo is going to be my working directory. So assuming that CDR is installed, you can then run this command, python-m cdr.bin.create_config. So the idea behind the implementation is that I've tried to automate everything as much as possible so that you can just run stuff using simple command line executables once you have a definition, a configuration file that describes the data that you're going to be using for fitting, as well as the kind of model that you want to fit. And then anything in this cdr.bin directory has a help that you can run by just putting -h. So if I run this -h, then it gives you usage instructions for the script. If I run create config -a, then it's just going to spit out a bunch of like a template for a configuration file for an experiment that I can then pipe into a file that I want to save it to. So I can call this here. This is going to be a synthetic data set, just because it fits quickly. So I'm going to call this synth.ini. So this is creating a new configuration file template for me to use at the path synth.ini. So if I open up that file, then I've got this kind of verbose description of all the different pieces of the config that I can use to set up my experiment. But there's really two critical sections of the config. The first is a data section that specifies where the data is that the model will load. And then the second is a model definition. So I'm going to go through the parts of this. Is everybody okay to move on? Does anybody need help with installation or data downloading? So in the data section, I minimally need a path to X stands for the predictors, so the stimuli. Oh, yeah. I hope. Yeah. Is that better? Yeah. All right. This is going to be a path to stimuli that I'm going to use in my experiment. So this would be, for example, I keep coming back to the reading example, but this might be the list of properties of the words as they were fixated in a reading experiment in order, for example. And then Y is going to refer to the path to the data that contains the responses that I want to model. So my experimental measure. Bold responses, EEG, reading, what have you. These are assumed to be in a tabular format. So if you don't have that, like a lot of neuroimaging preprocessing pipelines don't spit out CSV files, then it may require some kind of intervention in your pipeline in order to get a CSV that represents the data that you want to model. But this is kind of like, yeah, a standard assumption for these kinds of regression things, like in R, for example, that you just have a table that represents the data that you want to use for predictions and responses. And then you can specify some number of columns that indicate that the model will use to partition up the time series. So for example, if I have a separate time series for each participant in the experiment, and I have a column called participant, I might just put that in there. If I wanted it to be if I had a separate, multiple stories in my model or in my data, and I wanted a separate time series for each story for each participant, then I could do participant story with a space in between them to say I'm going to use both these columns to chunk up the time series. So whatever I give it, each unique value of the combination of these columns is going to, the model will treat it as a distinct time series, and everything else will be independent. So I'll delete this because we actually don't need this in the synthetic example. And then for the X train, the path that we're going to use is data, X, what did I call it? X.ev measures. And then Y train is going to be data, y.ev measures. So if you look at these files, if you just open them in a text editor, it's a space delimited tabular file that says what, and this is all synthesized, so these are not real. This is just generated programmatically, but what the values are for these, in this case, 20 different predictors labeled A through T. So these are going to be kind of simulated events in my experiment, along with the timestamps, for example, associated with them. And then if I look at the y.ev measures, it's the same. Only here I have this distinguished variable y that I'm going to be using as my response variable, as well as the time that y was sampled. The other predictors here you can just ignore. Yeah, so I've defined my paths to data. And then here, some things to keep in mind. So for example, if I want to filter my data in any way, remove outliers, I can specify filters here. I'm not going to do any filtering, so I can delete that. And then here, the separator in these tables is a space, but if it's like a comma, for example, I could do this. So just know how your data are formatted. And then output directory can be whatever you want. You just write a path to wherever you want the model to dump all of its outputs. So here we can just call it synth. So it'll create a new directory called synth, and it'll put everything in there. Yeah. Oh, and then this optional part that I skipped over on accident about you can also specify dev and test partitions of your data. Here we will actually not worry about that as well. So you can either leave it there or delete it. Yeah, so history length is the number of preceding events that will be considered. So although in theory, our convolution can operate over the entire preceding time series, it's pretty computationally inefficient to actually implement it that way, and so we cut it off at some time point. So here it's like no more than 128 things into the past.
Quick question from online. What is the role of history length here?
Yeah, sorry, that's the one I was just answering. So the no worries. Yep. So it's just a truncation of the history. So you should pick a generous value, like beyond which you don't think there will be any influences. All right.
One more quick one, Cory.
Mm-hmm.
What would the format be for the dev and test set tables if we were using them?
Yeah, it would be the same. Yeah, the config assumes that your data are formatted in the same way. So you would want kind of like a tabular representation with all of the predictors that you have used in your model in the xdev or xtest paths, and then all of the responses that you've modeled in the ydev and ytest paths. Yeah, if you're missing anything, the model will just complain that you don't have it, and then you can figure it out. And then, as I said, everything needs to be timestamped. So there's this distinguished variable called lowercase time that's expected in both the predictor and the response files. So you'll get a complaint if your data don't have that. OK, so the fun begins here at where you define the model. So here, you tell it that it's a model section by using this prefix model underscore. You tell it that it's a CDR model by using CDR underscore. Or not even underscore. So like that minimally, like if you want to define a CDR model, the section header needs to be this. And then you can define as many models as you want to of the same data. So you can, but they have to have unique names. So like I could have like, model 1 or 1, can't type. And then like another one down here that's 2, and so on. No constraints. You just need to have special names for them. So I pick names that are that you'll be able to interpret later. So here we can just call it CDR because we're just going to fit 1. And then the formula is kind of it's inspired by like linear modeling formula from R. So it has like a left-hand side and a right-hand side, where the left-hand side is the dependent variable. Then you have a tilde delimiter, and then the right-hand side is the predictors that you're going to be using for the modeling. So in this case, if you go back to the CDR tutorial, I've put a section called synth model formula. You can just copy that and paste that into the formula equals section here. And I'm jumping ahead to this so that we can get models running for anybody who wants to on your computers, and then I'm going to parse this for everyone. So once we've done that, assuming everything is configured correctly, we can go to our command line and type python mcdr.bin.train. And then we need the path to the config file, so synth.ini. And then you can do this little -m argument to tell it which model you want. So here we've called our model CDR. So that's what we'll do. If you don't tell it the model, it'll just run everything, all the models in the config file in sequence, which since we only have one is a viable option. But if you want to specify which model you want to run, this is how. All right. It seems like it's running. Good. All right, so it's just going to go through. An iteration represents a single pass through the entire data set. So it's going to run a bunch of these until it reaches its convergence criteria, and then it will stop. With this one, I expect this will, I don't know, probably take several hundred iterations. But so for if you've got a reasonably fast computer, maybe it'll be done by the time that this session's over. But it spits out some kind of useful stuff along the way, including like what its loss is. So the loss is going to be roughly proportional to the negative log likelihood of the model, along with some regularization terms if you have any, including in this case, it's by default a variational Bayesian model, so it also has a KL divergence regularizer. And then if you want to see the model's learning curves, which can be useful, you can just run a utility that's provided by the underlying library I'm using here called TensorFlow. So python-m tensorboard.main, and then log_dir equals, and then you want to use the path to your model. So in this case, it's going to be synth, I can't type, CDR. So this is where the model is saving. So we said already that our output directory is synth, and then each unique model name gets its own subdirectory there. So if I do that, then I should be able to pull up TensorBoard. Oh, I may have another instance of it running already. I do. That's the problem. There we go. This gives me some idea of the kind of trajectory of some of the parameter estimates in the model, which I'm not going to interpret for now. It gives me convergence criteria. So basically, as all these lines go up, I'm getting closer to converged. And then the learning curves are going to be here. So this loss by iter is going to tell me how my losses are changing with each iteration. So as that model runs, it will eventually spit out some logs. So I think it's going to probably do it every 100 iterations. So in a few seconds, we'll get some information about the model's fit. And it's going to get, and one of those logs that's of interest is it's going to plot the estimated impulse responses at this point in training. So you get those incrementally and can kind of check, because some of these models are long running, and you don't want to have to wait until the very end to see if there's something weird going on. But meanwhile, I thought it would be useful to actually look at some fitted models, and that's where I gave you the model, the models file as well. So while your synth model is training, you can go ahead and we can do some other stuff with the previously fitted models. So if we look at the models directory, again, there's going to be 3 different data sets, or models fitted to different data sets. And I'll just show the Fmri data set, since I think it's probably going to be familiar to the largest number of people here. So if you look at the contents of the Fmri data set, there's one model there. It's called cdr.b1024. And it also has a config file. So if you want to see kind of how that config file is structured, you can look at that. But it's the same basic setup as the one we looked at. It's just got some extra hyperparameters specified for the model. So if we want to plot what the model has estimated, then we can do cdr.bin.plot, and then give it the path to the Fmri model. So it's going to be models, Fmri, CDR. If we run that, it will produce some impulse response plots. Oh, CDR_B1024. So sorry, that's the name of the model. No, I'm losing my mind. It's the config. We give it a pointer to the config file, and then we tell it which model we want to run, which is CDR_B1024. So to review, to plot, we're going to run cdr.bin.plot. We give it the path to the config that defines the model, and then we tell it, using the -m argument, which model we want to run, which is named CDR_B1024. And then if we go to that location, eventually, once it's done plotting, there will be some PNGs that get spit out here that will tell us what the model's estimates look like for this domain. So I'll blow this up. So these are impulse response functions to different variables in a naturalistic fMRI data set where people listen to stories. So basically what you're seeing is the change in bold response on the y-axis associated with some delay after observing a word, so after the onset of a word in the auditory stimulus on the x-axis. So 0 represents no delay, like the instant of word onset. 5 represents 5 seconds after word onset. 10 is 10 seconds after word onset, and so on. So as you can see, this is roughly the same shape as the HRF that I kind of cartooned for you during the presentation, where you get a little response immediately, but then you get this kind of peaking primary response that happens around 5 seconds after the word is presented, which then dips, and then in the case of several variables at least, becomes a negative undershoot. So we actually get this kind of like, we've gone too far in delivering blood to this region, so we're actually going to dip. And then you get the response broken down by which predictor we're looking at. So we get a distinct predictor for rate. Rate is essentially an intercept term for this model, which represents the generic response to an event, regardless of its properties. So like, if a word happens, how will blood oxygenation in language selective areas change, regardless of what that word is, like in the average case. And then these other responses reflect deviation from that. So how much more or less will the response change if that word is more surprising than the average, or if that word has higher like acoustic loudness in the auditory stimulus. Right, and then so there's each of these predictors actually has its individual set of parameters that are going to describe not only the shape but also the amplitude of its response. So I can get a differential kind of sensitivity over time and different shaped responses to different kinds of predictors. Now, it happens to be the case in this domain that, again, as we said earlier, the response is dominated by these hemodynamics, which are largely shared across the different predictors. So we get very similar looking responses that vary only mostly in their amplitude. That won't be the case with other kinds of things.
Are shared. I wonder how you would differentiate whether like a response has to do with, let's say, how loud it is versus how surprising it is, because a surprising word can also be loud, and vice versa. Right,
and so the assumption, so one assumption you can make is that these are going to be, these effects are going to be additive, so that whatever contribution surprise on the one hand and loudness on the other make, they get summed together. So they're in kind of a zero sum with each other in the model. That would be like a standard assumption from linear modeling as well, or any kind of like hemodynamic response modeling will typically make that assumption. The model I'm showing here actually doesn't make that assumption because it's fitted with a neural net that can actually allow kind of saturating effects of both things, or nonlinear effects. And so for that, we actually, in this context, if we want to test whether these things make separate contributions, then we might want to fit like a null model that constrains the two to be additive with each other, for example. Yeah, in this case, right, we're just visualizing changes over time. But we can also visualize changes on the predictor dimension as well. So here we're looking at the temporal dimension, but we can also look at, in cases where we fit a nonlinear response function, we can look at what that nonlinearity looks like in the surprisal domain, for example, or in the sound power domain. So however we choose to define the model is going to determine the flexibility with which our model can adapt to these different aspects of the response function. So I think it would be confusing to turn there because it's kind of like jumping too much too fast. So for now, I think I'll instead show like how we might define the model in order to kind of enforce or relax different kinds of assumptions about the way it works. So I'll return to our synth model here. And I'll first show you kind of what a typical, I guess, linear model might look like. So not one with any convolutional structure. So we'll call this LM. if we just delete some of these terms, then we just have this 20 predictor linear model of our synthetic data, right? So here we're just looking at what are the contributions of all these predictors separately to y. This is a valid model in CDR. CDR will assume that anything that doesn't have a specified response function will use what's called a Dirac delta response function, which is just basically like a response function that's 1 at the same time and 0 elsewhere. So we're going to delete the contributions of all surrounding context except for stuff that occurs at the same time that we're measuring the response. So that reduces to a linear model of the predictors on the response. This isn't a good way of fitting such a model. You can do so a lot more efficiently in R, so this would not be a recommendation, but just kind of like to get a concept for how this works, then that is, these kinds of linear effects are possible to fit. If we want to add in convolutional structure, sorry, I should still need to keep this called CDR. CDR conv. Then we need this, this C term. So C is just like a 2-parameter function in the syntax of these formulas, where the first parameter is the set of predictors, and the second parameter is the response family. Mm-hmm.
Are those predictors?
Yeah. Yeah, so the right-hand side is going to describe all the predictors. And then in this context, we additionally describe not only which predictors we want to attend to, but what kinds of functions we want the model to be able to fit of those predictors in describing the response. So for example, if I do like, here, let's do this here. If I do this, then I've got these 21 different predictors, and then I have a response function that's a neural network. So take a neural net and fit the response to these predictors. I can constrain it to be something more specific if I have some reason to do so. So for example, I might want it to be in the family of double gamma hemodynamic response functions. So I could replace it with HRF if I know that I'm fitting Fmri data and I want to take advantage of prior knowledge about that domain. Or in the case of reading, I might assume that we've got a response that's going to be maximal at the current word and decay over time, and it's just a question of how steep that decay is. And so I might fit like an exponential family with like a 1-parameter exponential response function. The software implements several response functions, and if you're interested in using one that's not available, I'm happy to try to help you implement it. But yeah, the most flexible by far would be NN. Neural nets are universal function approximators, so you should probably get something close to what you need using this. Yeah, and so so I can actually specify like different response functions for different predictors. So I might say I want these predictors to have like an exponential shape, but these I don't know about. So I'm going to give them like a neural net shape or something like that. I can actually also specify fixed values for my predictors if I don't want them to be estimated. So in this case, here are the predictor values that are assumed by, say, SPM for Fmri modeling. So a double gamma hemodynamic response that has a 5-parameter shape with these fixed values. If I specify those values in my model, then this is equivalent to just pre-convolving with the canonical HRF and then fitting a linear model to that. So again, not something you would actually want to do, because this would be a way more computationally intensive way of doing it. But just a demonstration of the fact that we can fit familiar kinds of models within this space, but then we can also relax them just by deleting, for example, this parameter, in which case C, which represents the ratio between the overshoot, the primary response, and the undershoot component, would be fitted by the model rather than specified in advance. There are quite a few other things that can be done. I think since Ethan asked about it, I'll also mention that we can tie parameters by specifying a name for our impulse response function. So if we say iref_id, that's the name for the tire, that's the keyword argument for tying parameters, equals something. Let's call it, I don't know, single href. Then I'm going to fit one href, so one 5-parameter response function that will be applied to all of my predictors in the same way. And then my predictors will only be able to vary in the amplitude of their response to that function. So this is a useful thing for MRI where, again, in most cases, this is a reasonable assumption to make that there's just one response in a given brain region whose amplitude is modulated by your experiment. Yeah, and then the other kind of like thing to, that I think is going to be widely used would be mixed models. So here I've only specified everything at like a population level, so I'm assuming that the effects are going to be the same for all kind of entities in my experiment, but that may not necessarily be true. The kind of traditional syntax for mixed models in R would be like A plus B plus A plus B given ran F. So if you've ever fit models like this before, this means like I have population slopes for variables A and B, as well as random deviations of both of those slopes for each level of some random grouping factor. So this may be like a participant, for example. So I'm going to allow each participant's slopes for A and B to differ. Only it doesn't just have to apply to slopes or magnitudes. It can also apply to the parameters of their responses. They may have differently shaped responses. So for that, we just need to put the whole convolution call in the random effects component. So here we're fitting like a neural net IRF of A and B. And here we're allowing the parameters of that neural net to differ for each participant in the model. So this falls back to the population level estimate if you predict on data from participants who are unseen in training. So you should be able to use it kind of gracefully on any kind of prediction problem, even for new data. I guess I'll open it up for any practical questions. Did anybody's synth data finish? I can show you. I don't think mine did, but I can show you like where things stand with it. Yeah, so this is roughly what our response function looks like on the data that my computer is fitting so far. And then if I pull up the true response, nope, not there, here, you can see that it's relatively similar. The way that it's plotting with this legend is causing it to squish the y-axis, so it looks less steep, but it's getting pretty close to finding what it should. Right, in a completely simulated data set.
Yeah, and of some of these start at 0, and which those are just. I mean, in real data, we expect to have some.
Yeah, it's a good question, and yeah, so certainly that wouldn't be a good model of say ERP data. For other kinds of things like in the reading domain, these things do make sense because like a word will have some influence of its own properties. Or at least it could in principle. Yeah. Right. So yeah, so you can use, if you know that there should be no response at time 0, then you can use a kernel that will disallow that. So you could use, for example, like a gamma kernel. Yeah, this is just to demonstrate the flexibility that you don't have to make that assumption. You can fit whatever kind of response function you might need to. I thank you very much, all of you, for your attention. I know there was like kind of a lot here, but I'm very available by email to chat with anybody who wants to use this. So I will be your personal like help service because I would love to see some uptake. So thank you very much.